Hi everyone, welcome to Science Explained. I'm Rouah-Martin, Doctor of Science in Chemistry and Biotechnologies, and today we're going to talk about rapid tests and biosensors, and we're going to see how they work. Rapid tests and biosensors allow the fast detection of specific compounds on site without the use of any analytical equipment. There are different types of rapid tests and biosensors. So first, there are chemical tests, which are usually based on a chemical reaction, such as in the ethanol test. Then there are tests based on biological recognition elements, such as enzymes, antibodies, or DNA fragments. I will describe the glucose meter, which is based on an enzyme. Then we're going to see the pregnancy test kit, which is based on antibodies. And then finally, we're going to see a sensing solution for ergot alkaloids, which is based on DNA aptamers. The first sensors that were made were chemical sensors. They are usually based on a chemical reaction. I will give the example of the breath alcohol test or breathalyzer. This test involves a chemical dichromate ion, Cr2O7, which is orange, and which reacts with ethanol in order to form acetic acid, and chromium ion, Cr3+, which is green. The color change from orange to green indicates an excess of alcohol. This test is efficient, however, it can lack specificity, so it sometimes happens that some other volatile compounds present in the breath react with dichromate ion. So in case of uncertainty, a complementary test using analytical equipment can be needed. In order to improve the specificity of the sensor, it is possible to use a biological recognition element, such as an antibody, a DNA fragment, or an enzyme. And then, depending on the type of response we want to obtain, we're going to use different types of transducers. So for instance, if we want to obtain a color change, we're going to use an optical transducer. In case that we want to have an accurate measurement of the compound to be detected, then we're going to use an electrochemical transducer. And then in case that we would like to have an alarm beeping, then we're going to use an acoustic transducer. So depending on the type of compound to be detected, depending on the environment and the way we want to do the sampling, we can use different types of recognition elements linked to different types of transducers. The first sensor using a biological recognition element that was made was based on enzymes. It is a biosensor that is used by people having diabetes so that they can determine the amount of glucose they have in the blood. So most versions of glucose meters are based on an enzyme, the glucose oxidase, which produces gluconic acid and hydrogen peroxide, which is detected by an electrode. So here you have the chemical reaction that occurs. Glucose plus oxygen turn into gluconic acid plus hydrogen peroxide, H2O2. Then the electrochemical transducer allows the quantification of the concentration of glucose. So in order to use this device, the person puts a drop of blood onto a pad. The pad is then inserted into a reader, and then it will give an accurate measurement of the concentration of glucose in blood. So enzymes are very interesting recognition elements. They are very efficient for specific sensing. They have a few disadvantages, such as the fact that they are proteins, so that they can be degraded by certain chemical agents, for instance, and then it's not always possible to obtain an enzyme for the target molecule. Some biosensors are based on antibodies. I will give the example of the pregnancy test kit, which is based on antibodies. You have the scheme of an antibody on top, where are represented the heavy chains, light chains, and the antigen binding fragment. The antigen here is human growth hormone, HCG. So in this test, monoclonal antibodies are attached to the pregnancy test stick. If the person is pregnant, HCG that is present in the urine will bind to the monoclonal antibodies on the test stick. This allows a change in color or pattern by using a chromophore, and then it can indicate pregnancy. So if there's only one band, the person is not pregnant, and if there are two bands, there's the presence of AHCG, which will indicate that the person is pregnant. Antibodies can be very efficient recognition elements. They work really well for this kind of tests. However, they suffer from some disadvantages. So first, there are proteins, which can be easily degraded. Then they are produced in animals, so besides the fact that it's not ethical, it's sometimes difficult to produce on a large scale. And another problem is that many compounds do not generate any specific immunity. So it's not always possible to produce antibodies for every compound to be detected. The last type of biosensors I'm going to present is biosensors based on DNA aptamers. So first, the name aptamer, it comes from the Latin aptus, which means to fit, and the Greek meros, which means part. They are oligonucleotide or peptide molecules that bind to a specific target molecule. Then on top you have a 2D representation of a DNA aptamer, which is made of single-stranded nucleic acid, which folds on itself, and that gives it a particular shape so that it can very specifically recognize a target molecule and bind to it. Then you have the 3D representation of a DNA aptamer, which has two binding sites and which binds to the target molecule on ergot alkaloid. DNA aptamers can be linked to different types of transducers. In this case, DNA aptamers were linked to gold nanoparticles. So the solution of DNA and gold nanoparticles is pink, and then in presence of the target molecule, it turns into a dark purple precipitate. Aptamers have many advantages. So first, DNA is a very stable molecule. Then the synthetic production is ethical and it allows the production of large amounts of aptamers. Then there's the possibility of engineering the DNA aptamer so that it can recognize different molecules. Then it can be functionalized in order to be linked to different types of transducers. And then the high specificity and low interference with other compounds make DNA aptamers ideal candidates for the production of biosensors. So that was it. If you're interested about the topic of biosensors, I put a link to a review in the description.